So the next talk, as you saw from your programme, is by Robert Asher here, who's working in the museum here on mammalian paleobiology in Cambridge. He's done research in more out-of-the-way countries than even I have, which is quite good, actually. Geology is good, it takes you all over the world, but I think you've been to Argentina, Britain, Canada, Kenya, Madagascar, Mongolia, South Africa, Spain, the United States and Venezuela, you told us, and probably some others as well. Unlike the last speaker, who was a Brit working in America, Robert is an American working in Britain, so that's to keep parity. He did his degree in... actually, I don't know where you did your degree. Where did you do your degree?
PhD in Stony Brook University.
In Stony Brook, right. And then you've been at various places till you became curator of vertebrates here in the Museum of Zoology. And if you get a chance to go to that museum, it is a brilliant museum. Really fun to go around, isn't it? Well, I'm sure you think it is, but I think it is too. So it's probably not open on a Sunday, though, is it?
I regret to say it is not. Saturdays, though.
It's Saturday, so yeah. Well, I shouldn't encourage you to skip here and go there, but there you go, it is open. So I'm going to pass over to Robert to talk about how you construct evolutionary history from genetics and paleontology. So thank you.
Thank you. So I guess the first thing I'd like to do is kind of apologize for this ridiculously long reading list and text that has ended up in all of your programs. But on the plus side, there's slightly less reason for all of you to buy my book, because I put most of it in the handout. Okay, so there's really three major components to my talk today. As I'm writing here, I want to spend some time comparing trees, evolutionary trees of the sort that Ernst Haeckel published in 1874 here. I want to compare trees such as that were made in the 19th century with those that we look at today and explore with you what that means for the meaning of a tree and of the idea that animals and plants alive today share a common ancestry. I want to then talk about what those trees can help us, or how they can help us understand fossil genes, because I am a paleontologist, and while at first glance you might think that genes can be fossils, they actually can, and I think the level of competency in this room actually is quite high, and so that's not news, I don't think, to very many of you. And then finally I want to discuss a bit about how trees are relevant to fossil succession and how the stratigraphic record can help us understand the mechanism that Charles Darwin proposed 150 years ago as indeed a very compelling mechanism to explain biodiversity. So the bulk, pretty much everything I'm going to talk to you about is, relates to my profession as a paleobiologist. But of course this is a course on God and genetics, and so the very last slide I have some sort of questions for discussion, so we'll touch upon some of the several theological implications, I hope, in the discussion. Okay, so this is a tree that I presume all of you recognize. It's from one of Darwin's notebooks in the 1830s, and probably, at least in terms of a tree that actually draws upon biological mechanisms that we accept today, namely selection, this is the first tree diagram that represents common ancestry. And all it means is that we have living things, organisms today that exhibit variation. The basis of that variation is heritable, and you add those two things together along with some time, and the product of, or the sum of all of those three categories is evolution. So it's a fairly straightforward equation. And it has many parallels, and I think a good way to think about biological evolution is to compare it with linguistic evolution, because you can draw trees between many different things. It doesn't actually, drawing lines between points on a diagram by itself doesn't really mean very much. But I hope I can convince you with this parallel with languages that it does mean something very important about history. So what I'm showing you here are some Germanic languages, these living Germanic languages, Dutch, Afrikaans, English, and German. Here's a living Romance language, and they share ancestors that we could name them, and I'm sure the linguists among you may disagree with some of my choices of ancestors, but I hope you get the point. And this tree diagram is drawing on the same mechanisms of variation plus heritability plus time equals evolution, because there actually was a common ancestor of all Germanic languages that real human beings spoke 2,000-ish years ago, and I'd happily be corrected on that number by the linguists among you. So these are real languages that people spoke, and the mechanisms that that all of us see today in terms of a child growing up learning vocabulary and sentence structure, and all of you growing up learning those things, that kind of process that we observe today, variation plus heritability plus time, equals the evolution of languages. So what I want to do in the next couple of slides is explore this metaphor with linguistics a bit. And to start doing that, I don't have very much audience participation, but I have a little bit. So I want to sort of ask a question to all of you. Can someone tell me something that Germanic languages do that no Romance languages does in terms of grammar, sentence structure? And I know that's a very general kind of thing. I can give you a hint, but that'll probably give it away. Is there any guesses about what a Germanic language does that you can immediately distinguish it from a Romance language? Yes, it has to do with verbs, and it has to do with where you put them.
Second idea. Unless verb is second or after the subject, unless it's in a clause and then it goes at the end.
Well, both of you are quite close. It's not quite what I had in mind. I guess I'll just tell you then, unless you want to keep guessing. Germanic languages modify verbs with prepositions. So if you consider the sentence, I look the word up. Try saying, I look the word out, and it means something completely different. I watch up for traffic? No, I watch out for traffic. And Romance languages do not do that. And if someone, I'm still waiting for a linguist to tell me, oh yes, the Romanian does that. Okay, but I've yet to hear that. So what I've just described, this sort of morphology of living Germanic languages relative to a Romance language, is something that helps us build this tree. Even in the absence of any historical data about the people who lived 1,000 years ago or the things that they wrote, even without that, we have some data with which to build this tree of life. So this tree of life is telling us something about time. The ancestors that I'm showing you here, Middle Dutch, Old Saxon, Old Germanic, Latin, they precede the descendants in time. According to their position on this tree, you can say something about what's younger and what's older, which is not to say that the rates are all the same. The rate at which English has changed from its common ancestor with Dutch, the rate that English has changed from Old Saxon is not at all necessarily the same rate as how Dutch has changed from Old Saxon. That's different. But we can say that both Dutch and English, as they're spoken today, are younger languages than Old Saxon. And similarly, the close relatives on this tree, Dutch and Afrikaans, for example, should be more similar to one another than they are with their more distant relatives. And this modifying verbs with prepositions is an example of that. Which is not to say that you can't get some sort of cross-pollination, as it were, if I may switch my linguistic biological metaphors yet again. And English is a great example because, of course, Britain was occupied by the French for several hundred years, by the Normans. And we say things in English that sound pretty much like French. Beef, serve, history. The equivalents in German, Fleisch, dienen, Geschichte, do not sound one bit like these English words. And this is, of course, a reflection of the Norman heritage of us native English speakers. And so what I've done here on the tree is put little red hashes indicating we have some convergence, we have some parallelism. We have some, I suppose the real appropriate metaphor would be lateral gene transfer, right? We have some languages that are kind of infecting each other from, even though they don't share immediate common ancestry. So as long as you're aware of the pitfalls and the possibility of similarity occurring for reasons besides common ancestry, if you combine your awareness of the Norman history of English, what Germanic verbs do, no Romance languages do, you can still come up with this hypothesis of languages. Okay, so that's just using the morphology of living languages. And of course there is more data that we can use to reconstruct the tree of linguistic history, because these people who lived in the 9th century before the Normans wrote stuff down. We can find it, and we can read it. And this is a passage that I found on someone's apparently selling a copy of King Alfred's Orosius, which was written in the 9th century, somewhere in Oxfordshire. And I don't suppose anyone, I don't know, is there anyone who speaks Middle English? But this is apparently Middle English or Saxon, and it's an account from King Alfred's Orosius, and what I've done here is written out this one particular sentence. Here's the first one in this Middle English, Ac him heften pene poneveg forsetten, and that means, but to him had Carthaginians the road blocked, right? In the middle here I have the German, the modern German, aber ihm hatten Karthager den Weg verlegt. And in terms of the sentence structure, the German is very, very close to this Middle English, whereas the English is quite a bit different than both of them. In any event, the fact that we have a very Germanic sentence structure down here on this lineage leading towards modern English supports our hypothesis of interrelationships, using a bit of the fossil record of the English language, as it were. Okay, so this talk is actually not about linguistics, it's about biology. And I hope it's clear, though, why I told you that little soliloquy about languages, because it's very similar to biology. There are lots of sources of data that we can use from the natural world to demonstrate that descent with modification, mostly by the process of natural selection, is the means by which biodiversity today has evolved. We have comparative embryology, geographic distribution of organisms, some little evo-devo. We have patterns of relationships using different data sources and sequence of fossils and stratigraphy, and these two things are what the rest of the talk is about. And just to give you my conclusion in advance, looking at the morphology of languages is precisely like looking at the morphology of living animals and plants, constructing a tree. The fossil record is precisely like finding a copy of King Alfred's Orosius and asking, does this fit our pattern of interrelationships? And of course, molecular data are relevant to this too. So let me start then by sharing with you a tree. This is a tree that I drew, but it's based very closely on a quote from Thomas Henry Huxley's book, written in 1863, Man's Place in Nature, pages 80 to 81. And he just says in this quote that snakes and lizards, based on embryology, are very close to one another. Cats and dogs are very close to one another. And apes, chimps, gorillas, and humans are very close to one another in terms of their embryology. So all I've done is taken his text and converted it into a tree or cladogram, labeled the tips and some of the ancestral nodes here in precisely the same way that I did European languages a moment ago. Now, he could have drawn that tree. He could have said something like, the embryos of a lizard and a chimp are very close to one another. He didn't have to say the embryos of a lizard and a snake are close to one another. In fact, if you take this tree of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different species, how many different ways could we construct that? Well, here's a formula that will help you answer that question. If you replace the n with the number of species, in this case 10, you get this number, 34,459,425. There are 34 million ways in which you could draw a rooted bifurcating tree for 10 species. And he picked that one. He could have picked 34 million other ones, but he didn't. He picked that one. Here's another example of a 19th century tree. This one published in 1872 by Theodore Gill. This has a few more terminals on it. A few of them are not well resolved, so that's a sturgeon. And sturgeons ossify. They're actinopterygian fish. But they ossify their bones in a somewhat strange way. So in 1872, they weren't really sure what kind of animal that was. So that's not well resolved. That's a fossil jawless fish, and it wasn't known by a lot of stuff in the 1870s, so he didn't really know where that went. But he did say that Artelios are up here with other actinops like bichirs and garfish. He did know enough to say that our tetrapods, like mammals and birds and reptiles and amphibians, are close to lungfish and to coelacanths, which were known from fossils. They, in the 1870s, it was thought to be a completely extinct group. But of course, someone found one off the coast of Madagascar in the 1930s, and they were really surprised when they found a coelacanth because actually they had ordered the red snapper. So he's placing our lungfish and our coelacanths along with our tetrapods in a sarcopterygian clade. He has here cartilaginous fish, that's holocephalans, not very well known, but so-called bat fish. Here are rays and our sharks. Here, Theodore Gill in 1872 puts a clade of our lampreys and hagfish. And at the very base of our vertebrate tree, he puts branchiostoma, chordates. Now, a few of these things are a little bit disputed today. For example, it's generally regarded today that tunicates are closer to our crown vertebrates than our branchiostoma. Nevertheless, this is a pretty accurate tree diagram. And just to hit home that point, I want to plug in the number of, what else could he have said? Could he have said, for example, that a coelacanth is closer to a shark, or a hagfish is closer to a moray eel? He could have said those things, but he didn't. So I counted 18 terminals here that are resolved. If we take our n, replace it with 18, and solve this equation, we get a million, billion, trillion, quadrillion, 6.3 quintillion different possible rooted bifurcating trees that Theodore Gill could have drawn, but he didn't. He drew this one. Okay, so in the 21st century, actually for several decades now, we've had access to an extraordinary amount of data that neither Theodore Gill, nor Thomas Henry Huxley, nor Charles Darwin, or Ernst Haeckel, or any of these guys, they did not know these data existed in the mid-19th century. So you've heard it already, both last night and this morning, a bit about genetics. You know that we have the vast majority of our genetic information is located within the nucleus of a cell. This is a very cartoonish, unrealistic picture of a cell, but it does suffice to point out that that's a nucleus, and deep inside of it we have chromosomes, which are nothing more than tightly packed coils of this DNA molecule. And in a human being, we have 3.2 billion nucleotides, which actually compared to the number of potentially rooted bifurcating trees, or 18 species, is pretty small. But it's still a lot of genetic data. A mouse is 2.7 billion. This is a salamander, Necturus lewisi, and that has 120 billion nucleotides in its genome. Here's a teleos fish called a fugu with 400 million. So the actual size of the genomes across different animals vary tremendously, and actually in plants it's even more so. You can get 2 different species of onion that differ by an order of magnitude in terms of their genome size. I'm not really going to talk about genome size, but I think that's a really interesting observation. Anyways, all of these comprise data with which we can test ideas about evolutionary trees, much in the same way that we can test the linguistic history of English to test if it's a Germanic language. And as you heard last night, the nuclear genome is not the only genome that we have. We also have a mitochondrial genome. Here's my unrealistic picture of a cell. There's a mitochondrion. There's another one. Compared to the nuclear genome, the mitochondrial genome is very, very small. In a human being, there's only 13 coding genes, as Stephen clearly explained in the last talk. There are more non-coding genes, but most of those are these very small transfer RNAs that exist between the various coding segments. There are 2 pretty big non-coding genes, one for the large subunit ribosome and one for the small subunit ribosome in the mitochondrial genome. But just to remind you that of the size of this, the 20,000 bases in a human mitochondrial gene is 0.0006% of the size of a nuclear genome. So it's a teeny tiny bit of our diversity. So this is just a close-up of the circular mitochondrial genome. I'm not really going to talk about endosymbiosis, but it is said, and I think, as far as I know, it's a fairly widely accepted hypothesis, that we have a separate genome in all of our cells because a billion years ago there was a symbiotic event in evolution, so that this was some kind of bacterium-like creature that has left its signature in all of our genomes through an evolutionary event. So I want to now tell you a bit about the history of applying genetic data to build evolutionary trees of vertebrates. And back in the 90s, when I was a grad student, people were the mitochondrial genome is easier to sequence. Even though it's very, very small, there are more copies of it in any given cell. And for that reason, it's with the more primitive technology for sequencing that existed 20 years ago, it was easier to get mitochondrial information than it was nuclear. And there are 3 genes in particular that were widely used for vertebrates and mammals in particular. Cytochrome B was one of them, and the 2 ribosomal RNAs were the others. And here is just, I found some titles of papers from the 90s looking in particular at cytochrome B. Those are the days where if you found some obscure animal, these are 2 that I like a lot. That's a golden mole, an endemic African mammal. If any of you have know people with gardens in Cape Town, the things that their cats leave on the floor, as long as they don't have big teeth, because of course there are rodent moles in South Africa, but this is a golden mole. And if you could find a tissue sample for a golden mole, especially the more rare species, or of a tenrec, that's a microptamagale, that's an East African otter shrew, actually I beg your pardon, a West African otter shrew. If you can get tissue samples for these things, get a few loci cytochrome B, that's a high-profile journal publication in 1993, and that's what people where I did my postdoc were really, really concerned about getting obscure animals, getting genetic data for the cytochrome B, and building a tree for it. So, what I'm showing you below these 2 pictures is the GenBank accession for the cytochrome B gene of Eremetalpa grantee. That's Grant's golden mole, what I'm showing you here. And it's a very short gene. This is, in placental mammals, it almost always has exactly 1,140 nucleotides. And again, to put this in perspective, that one number, 1,140, is 0.00035% of the nuclear genome. So we're talking about a teeny little bit of genetic diversity. And one of the really fantastic things about molecular biology and about building trees in particular is that the level of data accessibility is really extraordinary. There's a very high standard. So if you publish a paper building a tree for your favorite obscure group of mammals, generally speaking, there are unfortunately a few exceptions, but generally speaking, you are obliged to upload all the sequences that you use to build that tree onto GenBank. And that's the website for GenBank. So many of you have heard about Ensemble. That's this ongoing genome project where there are about, from alpaca to zebrafish, there are about 60 or 70 different genome projects with varying levels of completeness. And all of those things are available via a drop-down menu on that website, ensemble.org. And I guess I'm fond of pointing out to people that, if you consider all the great life scientists of, the early 20th century, Linus Pauling, Francis Crick, just take all these people at the, in the 1950s or 60s, consider every unit of data that they ever produced or saw or even thought about, add them together, and anyone sitting here with a smartphone has more access to data than all those people put together. And that's because of this really extraordinary development in information accessibility. So one of the few things I did not put in that handout was a table in my book, available from fine bookstores everywhere, that tells you how to download sequences of cytochrome B for a number of different vertebrates from these public databases. You can follow the directions and download animals besides those that I downloaded, but this is what I did. It's table 1 in table 9.1 in figure 9.1 in my book. That's a fish, that's a frog, these are 2 birds, snakes, lizard, crocodile, opossum, cat, dog, orangutan, human. And if we just, remembering what Thomas Henry Huxley said in 1863 about who's closer to whom, you can test that using, for example, cytochrome B. You don't have to pick cytochrome B, you can pick some other gene because there's lots and lots of data on GenBank. And so, of course, I did that. I picked these particular animals and used a criterion. So I compared the sequences of these genes, and you could use a number of different optimality criteria. You can use raw similarity, for example. You count a match as a 0, you count a mismatch as a 1, and the animals that have the fewest differences are put next to each other on this tree. And here's the pattern of the tree pattern that results when you use a distance criterion. And just like Huxley said, here we have our cats and dogs together. Here we have our snakes closer to lizards than either is to a bird. Frogs are outside of our amniote clade. The fish is outside of our tetrapod clade. All of these statements that Huxley made are consistent with what I'm showing you here. Of course, those of you who are into vertebrate evolution will recognize that this is not actually correct in a few regards. For example, birds are actually more closely related to a crocodile than a lizard is related to a crocodile. And that results from the fact that this is a very quick and dirty optimality criterion, raw similarity, and it's also a teeny tiny bit of genetic information. So if we can address those issues in a couple ways. We can use a somewhat more sophisticated tree reconstruction optimality criterion for our cytochrome B sequence, and the one I'm showing you here on the right is parsimony. Parsimony is not quite the same as distance. Parsimony says that the optimal tree will minimize the amount of character change. It ultimately is this general principle in science. The simplest explanation is the best. And you can apply that in a very algorithmic way to differences in nucleotides and come up with a tree that puts, it's a little bit better than the distance one because it puts our snakes together with our lizards and crocodiles. However, parsimony doesn't do a good job with the birds, and that little squiggle is meant to tell you that the birds in this tree are actually outside of a diapsid mammal clade, and that is inaccurate. Of course, the reason that's inaccurate is because we're looking at 0.00035% of the nuclear genome. We're looking at 1,140 bases of Site B, which is a teeny tiny amount of data. And we can do much better, and of course many of our colleagues have done much better. So here's 3 papers that I've picked because they happen to have a largely overlapping set of species. There are many other papers that will tell you the same thing. This paper by Rafael Sardoya and colleagues in 2003 is pictured here on the left, and it shows a tree that's completely consistent with the statements made by Huxley in 1863. This is another paper by Hugel and colleague in 2007. They look at a nuclear gene called RAG1. It's completely consistent with the mitogenomic phylogeny. Frederick Delsuc, a colleague of mine from Montpellier, published a much, much larger analysis of 179 nuclear genes, still a very small component of the overall genetic diversity of these animals. Nevertheless, the tree shown here at the left is supported quite emphatically by all of these different papers, and it's consistent with what Huxley wrote, what Ernst Haeckel wrote, and what Theodore Gill wrote and indeed figured in their papers from the 1860s and 1870s. So molecular biology, the conclusion for this first part of the talk is that molecular biology has confirmed the basic structure of the vertebrate tree as proposed in the 19th century. Now, those of you who have followed phylogenetics and are interested in vertebrates and mammals in particular will want me to tell you that not everything, it's not simply validating stuff that people said in the 19th century. There are many, many surprises that have happened in the course of applying these really huge amounts of molecular data to building trees. And I'll just tell you a couple of these surprises. One of them started to become apparent in the early 90s, sort of overlapping when I was a grad student. And this is the idea that every whale that's alive today is not just closely related to artiodactyls. Artiodactyls are even-toed ungulates, so they carry the weight between digits 3 and 4 in the hand and the foot. Artiodactyls are things like deer and cows and camels and pigs and sheep and hippos. And as it turns out, any sequence study that you've ever seen, any decent sample of molecular data for whales and artiodactyls and other mammals will put the whales within Artiodactyla closer to a hippopotamus than that hippo is to, say, a pig or a camel or a cow. And that was quite shocking to a lot of the mammalian systematics community. And what I'm showing you here is what is more or less the standard for mammalian systematics for the last 10 years, as compared to a tree that came out that was state-of-the-art in the early 90s. And what you got back then was some of the higher, some of the associations of orders were accurately recognized back then. People knew that, say, for example, rabbits and rodents were closely related. People knew that, for example, sea cows and elephants were somewhere near each other. But there is still a lot of ambiguity in the support indices for these various deep nodes weren't high. And you could get another study that looked at some other system that would support something completely different. But the state of affairs today is that the more data we apply to these deep nodes, the more confidence we get in these four basic clades: the Narthrins, Afrotheres, Laertiotheres, Urocontibules. I guess I won't bore you with the details of the constituents of those groups. With a few exceptions, I'll tell you a little bit about Afrotheria because it's my favorite group. But I just want to convey to you the extent to which this really was surprising. So Afrotheria is this sort of dark blue clade here. And basically what Afrotheria is telling us, if you look at the overall phenotype of a golden mole from South Africa versus a European mole, there are some major differences, one of which, of course, is the way these animals dig. European moles use these big mitts, these hands, to dig in this kind of lateral fashion, whereas golden moles are digging in a much more parasagittal way. I mean, there are several other differences. Nevertheless, no one, up until the 1990s with the profusion of molecular data, would ever have suggested that actually this golden mole shares common ancestry with an elephant to the exclusion of this European mole. But it does. And the molecular data are unambiguous about that relationship. So that's another surprise. I guess, though, even with those surprises within the placental mammal radiations, if we compare this modern tree of mammals with the tree that Gill published in 1872, he didn't really say anything about how mammals are interrelated. He just sort of put them up there under one term. So I guess it's important to recognize that, the application of molecular data to the tree of life since the 19th century has not been static. It has resulted in some changes, but the basic structure of the vertebrate tree of life has remained remarkably stable, especially when you consider that there were, how many was that? 6.3 quintillion? I mean, all the different possible ways that you could arrange these 18 species. The range at which this is correct, the very narrow left margin of that big curve of alternative trees is truly remarkable. Okay, so that's the first part of the talk, and I hope I've made that clear, right? So what I've just done to summarize the first part of the talk is applied molecular data to the tree of life that was essentially built using comparative embryology, comparative anatomy, and it's very, very accurate. What I want to do now is justify I think there's a paleontology somewhere in my title, so I want to start on that paleontology side, and I'm going to do so in a kind of cheeky fashion in that my sort of paleontology initially has to do with fossil genes, because there are such things. And fossil genes we're capable of recognizing them because we have a good tree of mammalian evolution, for example, where I happen to work on mammals, so that's what I focus on. But so I want to start telling you a bit about fossil genes. And I want to use the interesting fact that some mammals have well-developed teeth and other mammals have no teeth at all. I want to use that to predict where should we find fossil genes in the mammalian tree of life. So I guess the dentists among you know that, at least in a human, a tooth is not a uniform structure. You have some very different tissues in it. You have enamel, the hardest substance synthesized by the body, and you have the other hard substance is, of course, dentin. You have cementum that anchors the root to the bone in the jaw. And the majority of mammals also have these structures in their teeth, enamel and dentin, but not all of them. So here I'm circling some groups. So a sloth and sloths and armadillos do have teeth, but they don't have any enamel. They have purely dentin-composed teeth, which are pretty hard, but not quite as hard as enamel. Anteaters, of course, completely lack teeth, none at all. Pangolins are the same. Where are pangolins? That's a pangolin way over here in the Laertiotheria part of the mammalian tree, completely edentulous. Baleen whales, a blue whale or a minke whale, has baleen. It's this keratinous substance, a bit like fingernail, and they have no teeth as adults. And of course, aardvarks, they're a bit like armadillos and sloths in that they have these weird concentrations of dentin as teeth, but they don't have enamel-covered teeth. Now, an interesting thing about the development of these toothless animals, or either toothless or enamel-less animals, is that early in their own development, you can actually see the rudiments of teeth forming. So this is a fetus of a finback whale, and what it's showing you here are the emerging tooth buds that at this particular point in development you can see them, but these, in a different animal, they would continue to develop, break the gum, and serve a function in the adult, but they don't serve any function at all. These are buds. They will disintegrate as the animal continues to develop, because of course baleen whales have baleen. They don't have any teeth at all. On a genetic level, there is a bit known about the genetic basis of tooth development, and there are three loci in particular that I want to mention to you: enamelin, ameloblastin, and dental matrix protein 1. The first two are known to be important for the development of enamel, for the synthesis of enamel as a tissue, and the last one is thought to be relevant to the formation of dentine and also to some extent of bone. Now, here's where we start talking about fossil genes. In order to appreciate what a fossil gene is, I suppose I'm obliged to define a few terms. And Stephen, in the last talk, did a bit of this work for me, but forgive me, I'll repeat a little bit about it. So basically, in order to get that information from the nucleus into a peptide, into a functional protein, you need two processes: transcription and translation. Transcription is when messenger RNA enters the nucleus, copies the information in that chromosome, brings it outside of the nucleus towards a structure called the ribosome, which I've shown you. Here's a cartoon of one, enabling the ribosome to initiate the process of translation. And translation is the process by which that genetic information, originally from the nucleus, temporarily on that messenger RNA molecule, is joining these transfer RNAs. These transfer RNAs, so that actual amino acids can be assembled together in the correct order, right? So a fossil gene is when somewhere, it could be happen during transcription or translation, somewhere in this process, there's an interruption. And one way in which you can get an interruption is in the sequence of the original coding DNA inside the nucleus, a base is deleted, because of course you need to have bases in triplets, in groups of 3, for a coding gene to ultimately result in a functional peptide, in a functional protein. So if you were to delete that G, that would shift everything downstream, so that the identity of that triplet, everything else downstream from that original mutation, is going to be off by one nucleotide, and that makes a huge difference. So that's called a frame shift mutation. Another kind of fossil gene is when you get a certain combination of nucleotides that spells out a stop codon, which tells the protein assembly machinery, of course, to stop. And so instead of a functional polypeptide, you get this partial thing that doesn't synthesize completely. So, having defined, I hope, fossil genes in some comprehensible way, I want to point out to you now this paper, this very interesting paper that was published in 2008 by Tom Demaray and colleagues. And they look specifically at the 3 loci in baleen whales and some near relatives. They looked at enamelin, ameloblastin, and dental matrix protein 1, and they asked, do we find pseudogenes in these enamel and tooth-relevant genes where we expect to, according to the tree? So here are all the baleen whales that, as adults, they don't have functional teeth. Here are a few tooth whales. That's a dolphin. That's a sperm whale. They, of course, do have functional teeth as adults. Here are some other artiodactyls. That's a pygmy hippopotamus. That's some suiforms, pigs, camel, llama. That's a horse. That's a carnivore. Some rodents and a human. And what they're showing you are either these little blue notches. They represent mutations in the coding region of one of these 3 genes. But the blue indicates that all of those mutations happen in sets of 3. So if you were to shift, say, for example, this codon 4, 3 nucleotides down and insert an extra protein, you wouldn't necessarily ruin the capacity of a functional peptide to result from that process. Or alternatively, you could delete something in a set of 3, and it would still preserve, in theory, the functionality of that gene. However, like I said, if you were to delete just one, you'd get what I defined before as a frame shift mutation, and that's what these red hash marks mean. And the take-home message, what Demaray et al. Are telling you, is that you don't see frame shift mutations in animals that have functional teeth. Here are the animals with functional teeth. You only see mutations in groups of 3. Here are the baleen whales. You only see, or the only times you do see frameshift mutations is in the animals with non-functional teeth in adults. Here's a very similar paper from Meredith et al. In 2009. They were focusing on just one of these genes relevant to teeth, that's enamelin. And it's a very similar message, except that they have a much bigger taxon sample. So they sampled baleen whales, and just as Desmarais et al. Found, they too find that you get frameshift mutations and stop codons within your baleen whale enamelin. They also had sequences of this gene for every other, at least one representative of every other group of mammals that have teeth. So that's an aardvark that lack enamel. Here we have a species of a toothed whale, dwarf sperm whales, that actually, they have teeth, but the enamel is poorly developed. That's a pangolin, they completely lack teeth, and here are our xenarthrans, our sloths and our armadillos, and they too completely lack, at least the sloths do, the armadillos completely lack enamel. And anywhere you see this red, purple, or blue is where you see the accumulation of these dental pseudogenes. I'm kind of belaboring this point. And I'll belabor it a little bit longer because it's not just teeth where you find pseudogenes. You can find it in genes that are relevant for smell, for olfaction sensitivity. Humans have an extremely small nose. Next time you're cuddling up next to your pet Labrador, consider the fact that most of that dog's head is consisting of nose, and yours is teeny tiny compared to that. So compared to most other vertebrates, human beings have a very small nose and a very high proportion of olfactory pseudogenes, because we depend much more on vision than we do olfaction. Of course, if you're an aquatic animal like whales and sea cows, smell in an aquatic environment is very different. It is demonstrated that aquatic mammals also have a very high proportion of olfactory pseudogenes. The same is true in animals that have lost their sense of vision. Pandas are the only vegetarian members of the order Carnivora, and the panda genome a couple years ago demonstrated that they have a pseudogene for a taste receptor that's sensitive to an amino acid that only occurs in muscle tissue. The list is long, and I guess the take-home message that I want to convey to you is that these pseudogenes are acting as signposts. They're telling us that a vegetarian panda descended from a carnivore, from a carnivoran that ate meat. They're telling us that baleen whales that lack enamel-covered teeth descended from mammals that did have teeth, and blind subterranean rodents descended from other species that could see. And here I give you a quote from Origin of Species, in which he's essentially outlining, he's making a prediction that we should, he's making the prediction in terms of vestigial organs, but it's equally applicable to pseudogenes. So sometimes you hear in sort of the anti-evolution crowd that a pseudogene is an example of suboptimal design, much in the same way that a Ford Pinto is an awful car. None of you should ever buy one. Even if you could. I suppose maybe there's one on eBay nowadays, but they're really bad cars. The fuel tank is right next to the exhaust, and if you get rear-ended by someone else, the Ford Pinto is likely to explode. It's an awful design, but it doesn't mean that it wasn't designed, and you'll find that argument made by a lot of people in the ID sympathetic crowd. But this is not about suboptimal design. Pseudogenes are not about suboptimal design. Pseudogenes occur in exactly the places you would expect, given the mammalian tree of life. We don't put all these toothless animals in a clade together. Based on other bits of information, we put them in clades with toothed animals, and we find the genetic rudiments that their common ancestors have. Okay, and so the other thing I want to very briefly touch on in the minutes I have left has to do with this ENCODE project, which really got a lot of publicity in the last there were about 30 papers in this this very highly orchestrated media event, and all of you heard some of the headlines, I'm sure. So 80% of the genome is functional, is what the very talented people in this ENCODE project said, and in strictly speaking, it's true. But I guess what I want to point out to you is what I'm describing to you as fossil DNA. I'm not saying that there's no function for a pseudogenic in the ameloblastic. I'm not saying that some of these dental enamel genes have no function whatsoever, because what the ENCODE project, the criterion that they used for functional was quite minimal, biochemical activity. So it's entirely possible, in fact it's very, very likely, that in a sperm whale, in a mystacete, in a baleen whale, one of those enamel relevant genes is still being synthesized by a messenger RNA being sent out into the cytoplasm of the cell. The status of a fossil gene does not preclude the transcriptional activity of that gene. And so by the ENCODE project standards, that whale, that baleen whale pseudogene enamelin gene could still be quote-unquote functional. So the really important message is still that these fossil genes are signposts in the tree of life, occurring exactly where you'd expect them to be, using natural selection as a mechanism by which all these animals evolved. And so I'm just going to, if anyone's interested, I've written down some blogging sites that I think give a lot of very important perspective on the results of the ENCODE project. And I don't expect anyone to scribble these URLs down, but they're in there so that you can ask me afterwards if you want the URLs, and I can just give it to you that way. Okay, I want to end my talk finally getting to real fossils, geological fossils, fossils that you can hold in your hand. And I want to start by pointing out another passage in The Origin of Species in which he's basically saying that we should expect to find in the geological record what often is referred to as missing links. And I'll just read to you a little bit of this. All living species have been connected with the parent species of each genus by differences not greater than we see between the varieties of the same species at the present day, and these parent species, now generally extinct, have in turn been similarly connected, and so on backwards, always converging to the common ancestor of each great class. So this statement is telling us that if we have information about past life, we should be able to find an animal that's intermediate between two living animals and the common ancestor that they share. So, going back to this vertebrate tree of life that I articulated in the very beginning of the tree, this is, of course, not from Gill, this is from Haeckel, and he articulates a vertebrate pattern that's essentially the same. So all vertebrates share a common ancestor, all jawed fish share a common ancestor, bony fish, tetrapods, synapsids, etc. And it's, if you remember, I said that the tree is telling you something about time. So we expect to find the oldest anatomical evidence for vertebrae, or for segmentation, to occur in an older point in geological history than we do, say, for example, jaws. Similarly, we expect to find the oldest evidence for habitual bipedalism in a point in the fossil record that's younger than the anatomical signature of being a primate. And there are a few, but one of them that you may or may not have heard of is having a middle ear space enclosed by a bone called the petrosal. And I'll show you a picture of that in a minute. And what I'm here to tell you today is something that I'm sure you already know. I mean, this is bleeding edge science from about 1908. Some of the details aren't. I mean, Sahelanthropus is actually a very recent addition to the hominin fossil record. In any event, Sahelanthropus is a late Miocene hominin. It's on the lineage close to humanity. We share a more recent common ancestor with this African fossil than we do with a chimpanzee or a gorilla or an orangutan. And the reason that this fossil is inferred to be habitually bipedal, at least in part, is that the foramen magnum is positioned very ventrally as opposed to posteriorly. Now, I'm picking Sahelanthropus because that's the oldest. There are other hominin fossils. That's really one of the better parts of the mammalian fossil record, and some of you are probably quite familiar with that already, but I could have picked Ardipithecus, I could have picked Australopithecus. There's a long list of hominin fossils that show clear evidence of habitual bipedalism, but you don't get that older than the late Miocene. It's late Miocene or younger. On the other hand, if you look at the anatomical signature of being a great ape, so living great apes are gibbons, orangutans, chimps, gorillas, and humans, what we have is a relatively short and robust lumbar region. What we don't have is a tail. Monkeys have tails. We do not have tails. This is a fossil of an animal called Morotopithecus from the Lake Victoria region in East Africa. And the skeptics among you might need some convincing, but you really can say a lot about vertebral anatomy based on a strategically found vertebrae. And this is a single lumbar vertebrae that it's not the only thing known for Merodopithecus. Merodopithecus is known from other fossils. Suffice it to say, though, there's a good case to say that Merodopithecus had a robust and short lumbar region and did not have a tail. And of course, there are younger great ape fossils, something like Dryopithecus, that is known from much better material, that is also older than our oldest habitually bipedal fossils. So the point I'm trying to say is that the order in which we find these fossils didn't have to be this way. There's, if evolution by natural selection didn't happen, we could have found the oldest evidence for bipedalism in the Devonian somewhere, but we don't. And let me give you just a couple more examples. Here's our Merodopithecus, like I just said. Shoshonius. This is an animal from a quarry that I worked at back in 1992 and 1993 called the Buck Springs Quarry in the middle of Wyoming. And you probably can't recognize what that is, but these are teeth. That's a zygomatic arch. That's a foramen magnum. This is a teeny little omomoid skull. It's a primate skull. And the composition of that auditory bulla, where your middle ear ossicles are housed, is contributed to substantially by the petrosal bone. And every living primate today has an auditory bulla enclosed by the petrosal bone. You don't need to know anything about the fossil record to recognize that a petrosal bulla is a signature feature of primates. And Shoshonius just happens to be 55 million years old, much older than Merodopithecus and much older than Sahelanthropus, fitting its position in this tree of life. And I think this is the last example like this that I have. Okay, so Shoshonius is our oldest. It's actually not the oldest primate. There are other indices of primates that are a bit older than Shoshonius, but not much. It's upper Paleocene, early Eocene. Nothing comes close in the primate record to the age of this fossil, Morganucodon. And here in Cambridge, we have perhaps the world's best collection of Morganucodon fossils. This is the geologically oldest mammaliform vertebrate. And we say it's a mammaliform because it's very clearly showing us a condyle for articulation with the skull that's formed by the dentary bone. So everyone in this room has one bone in their jaw, whereas a chicken or a crocodile or a snake has several. Furthermore, we have multiple bones in our middle ear space. We have Malleus, incus, and stapes, whereas a lizard has just one, a stapes or columella. That's a long story, something I can tell you about another time. Suffice it to say that exactly in correspondence to its position in the fossil record, according to its position in the tree of life, our oldest evidence for a mammaliform is much older than the oldest evidence for a primate. And the same thing is true for the oldest evidence of a whale, or for a kangaroo, or for an echidna, because all of those groups are in more nested categories than the mammaliforms. And again, it didn't have to be that way. What I'm telling you in these past couple slides is summed up in the phrase, there are no rabbits in the Devonian. In other words, we could have found this sequence of fossils in other ways, but we don't. And that, to me as a paleontologist, is a really, really important line of evidence in support of this prediction that Darwin made in 1859. So the details, what I've just shown you are nice pictures of things. I'm telling you how old they are. I can appreciate the skeptics among you may not be convinced, but I hope, if you're not convinced, that you'll look at a substantial literature from people like my former postdoc advisor, Chris Sidor, Jim Hobson, John Fernarelli, Will Clyde. These are all papers that are taking tree diagrams like this, saying that, okay, this node is supposed to be represented by fossils that are younger than this node. Is that true, according to the stratigraphic record? All these papers do this. There is noise. It's not you will find exceptions because fossilization has no necessary relationship to speciation. There's a perfectly legitimate reason why we should not expect a perfect correspondence between the two, but all of these papers support with statistical significance that, generally speaking, you do find this correlation. And let me just give you one example of noise that does occur in the fossil record, and that has to do with the evolution of elephants, which have a really great fossil record that tends to be less well appreciated by the public than, say, hominids or whales or I don't know, basal tetrapods. In any event, elephants have a great fossil record spanning almost 60 million years. And what we find in modern elephants are these huge gargantuan things Loxodonta. It's a pale remnant of this diversity that used to characterize the proboscidean radiation. Most of the taxa throughout the Oligocene, for example, are much smaller than living elephants, with this interesting exception. It's an Eocene North African fossil called Numiditherium. It's about the size of a horse. It's definitely smaller than a modern elephant, but that skull, a skull of a Numiditherium found in the mid-'80s by a French North African team, it's probably about that big. I've seen a cast of it in Emmanuel Geerbrand's lab in Paris. Probably about that big. And it's a bit odd because it has this heavily scarred by muscles skull. In other words, one of the reasons, or one of the ways in which we can infer the existence of a trunk, because if you're carrying around, 90 kilos of trunk on your head, you have to have the muscular infrastructure to do that. And that leaves very telltale signatures on the skull. And Numiditherium seems to have that kind of basis. Whereas if you look on some of these animals that are actually more closely related to a living elephant, they don't. They're much smaller. An animal like Moeritherium, known from the early Oligocene of North Africa, does not have evidence certainly not for a long trunk. In any event, this is an example of noise. It's something that you don't expect this finely graded sequence of, a little bit like a modern elephant, more like a modern elephant. You don't find that in the fossil record, and nor should you, for reasons that perhaps we can talk about during the discussion. Okay, so really that's the substance of what I have to say to you. I want to close with, again, this notebook sketch of a phylogenetic tree that Charles Darwin drew in the 1830s. I want to remind you that the kind of information that we have at our disposal to build evolutionary trees, whether it's from the mitochondrial genome, whether it's from the nuclear genome, whether it's from comparative embryology, the fact that all of these very, very different bodies of evidence lead to trees that are largely congruent, and keep in mind the vast number of possibilities that we have, 6.3 quadrillion different possibilities for, quintillion possibilities for only 18 species. The fact that you do get trees that overlap is spot on in terms of a Darwinian evolutionary perspective. And remember that this is no different than reconstructing the history of languages, using a process that all of us are familiar with, namely learning a language, making mistakes. Those mistakes get propagated and end up being, for example, American English, which is a series of mistakes that have built upon the language that most of you speak with such lovely accents, and I do not. So I've said nothing about theological implications, but there are some. And I'll close. This is a very drawn-out closure, isn't it? But the sort of theological implication, one of them has to do with this possibility. Does this mechanism, descent with modification, which that's the vanilla way of phrasing it. You could also say, nature red in tooth and claw, survival of the fittest. Those are not so vanilla ways of phrasing it. And does it not entail some level of suffering? And would a benevolent creator use a horrible mechanism such as natural selection to create biodiversity? That is something important to talk about. What you often hear in the sort of anti-Darwin crowd is that their views are being marginalized. Anyone who's seen that movie Expelled from 2008, I was compelled to buy a used copy of the DVD because I reviewed the movie for a newspaper a couple of years ago. It's truly an awful movie. But I'd love to tell you in great detail why I think it's truly an awful movie. And they make the claim that their, what they would say, perfectly legitimate views are being marginalized, they're being excluded, much like the Stasi in East Germany were excluding dissent. They try to make that case. That's something else to talk about. Another more explicitly theological implication has to do with the category of a supernatural explanation. Am I ruling out supernatural explanations for biodiversity when I tell you that you should accept the evidence for descent with modification as the mechanism by which biodiversity exists today? That's something to talk about. So I guess I really will end now with that, and I'll thank you very much for your attention.
Thank you, Robert. Well, that's thrown up some challenges. I don't know if anybody wants to address any of these three questions here, or indeed anything else from Robert's lecture. So let's start.
Well, I'll jump right in with theological questions. Based on the last talk and your talk, and the finding of genetic relationship between all the domains of life and that we're all using the same genetic code. What do you think that has to say about the notion of an imago Dei, the man made in the image of God? Does the fact that we're only 0.01% unique genetic species rule out any kind of imago Dei, or do you think that's enough?
I don't think it rules out, humanity. I guess so the short answer is no, I don't think that rules out a scriptural understanding of humanity created in the image of God. I guess I would qualify that by saying that what I've described to you is a mechanism. And so consider, and those of you who've heard me blabber on about this, forgive me, I'm going to repeat something that you've heard before, but consider the proposition that Thomas Edison made the first light bulb, and electric light functions by, a heated coil running through a vacuum. Neither one of those explanations is contingent upon the other. You can be intimately aware of the details of vacuums and combustion, and yet know nothing about Thomas Edison. Conversely, you can know everything about Thomas Edison and be completely ignorant about the mechanisms by which that thing worked. I find it very helpful to look at a mechanistic explanation of biodiversity in terms of, evolutionary biology, in terms of selection, as the means by which biodiversity exists, which is independent. It doesn't confirm or deny the possibility of some agency behind it. Now, are we created in the image of God? I think that question is more relevant to our capacity to recognize reason, to use it, and is that contingent on having 5 digits? Is that contingent on being a primate? I don't think so at all. So I guess you see this once in a while in the literature, people saying, well, if we were hyper-intelligent cephalopods, would we not be created in the image of God? I don't think that follows, but that's complete speculation on my part. So if we I disagree in some ways with Simon on this point. Well, he's not here to defend himself, so I guess I shouldn't use him as a straw man, but I get the sense that he sees it necessary for anthropoid primates to be the image of God, and he, again, that's perhaps a bit unfair. Personally, I think it's really the image of God, to me, personally, means the capacity to use and recognize reason, not having 5 fingers or habitual ebola or habitual bipedalism.
So if we were kind of elephants-like creatures that could reason, then obviously you would say
Which, incidentally, if you had to pick an animal that probably could reason, it is an elephant. I think. But I'm sorry, go ahead.
No, that's okay.
Sort of following on from that, from a theological point of view, I find the notion of the image of God quite problematic in terms of any kind of human creature or any other creature. It's very difficult for human beings to conceptualize disembodied consciousness, but impossible to conceptualize in ordinary terms. But that is, in effect, what the religions say. They don't really, the image of God is a metaphor. It's a language form. And I think this question, an explanation, mechanistic explanation, tells you how something works. That is absolutely fine and in many ways convincing. Or I'm not a scientist, but to me, just probably because I'm not, I find the explanation of how something has come about very acceptable. It doesn't address the why question, and that's where I see science and theology complementary in terms of that they are asking the questions. They're not. Theology doesn't talk about how as much as it talks about why.
I would agree with you, but I think that's a sticky point, and you will find some of our colleagues, perhaps those of you, some of you in this room, who do want, I don't know. It's kind of like a turf war, really, But I'm very sympathetic with what you said.
Right, let's go.
You mentioned the origin of many large clades of mammals arising. That they just time and time again you mentioned them, and they seem to be between the time of 50 to 60 million years old. So that gives me an excuse to ask the question I wanted to ask of you anyway, which is why do you think that the Eocene was the early Eocene is basically when all or many of the major groups of mammals arose?
Very good question. The answer to that question is somewhat contingent on our understanding of, because I mean, part of it is undoubtedly taphonomic. Part of it is undoubtedly the artifact of what parts of the Earth today are eroding. Huge tracts of the western interior of the United States and Canada. I don't know. You guys know what Wyoming looks like? You know, it's badlands. You don't find badlands in Britain. Of course, you do find lovely cliffs eroding. You do find fantastic river cuts. You know, but in terms of giving you access to a certain time and place in Earth history, that is a very biased sample. And so in terms of the early Eocene, you know, there's something about the fact that the Rockies have an uplift that started, you know, somewhere in the vicinity of the Upper Paleocene, that that part of the world is not covered by lush tropical rainforests. You know, if we had the Badlands around the equator, like I very, very much regret to say we probably will in another couple hundred years, you know, that the problem would be quite different. We would have access to outcrops in parts of the world that are very different than we have today. So partly the answer is preservational. What I started describing was using completely different, well, somewhat different strategies to infer when and to some extent where these common ancestors for mammalian orders, for rodents, for primates, for cetaceans, for percodactyls, etc., when did they actually exist? And of course, this is the molecular clock. You need fossils a little bit, but using a clock philosophy, or it's not really philosophy, using a clock methodology, you can say something about, you know, were there baleen whales in the Upper Cretaceous? And there happens to be a paper, Dos Reyes et al., to which I myself contributed, so I, of course, highly recommend it to you, published just a few months ago in Proceedings of Royal Society, applying the largest genome-based data set to mammalian divergence events. And basically, this, what often had been discussed in the literature as this kind of, you know, huge gap, based on other clock studies that had used much smaller data sets, had much wider error margins. Some of them were saying that you should find rodents 100 million years ago, but you don't. There's no paleontological basis at all for 100 million year old rodents. But once in Dos Reyes et al., the genome sample size got much, much bigger, and the gap has shrunk accordingly. And not only has the gap shrunk, but the error margins has shrunk too. So that, I guess, that's not really answering your question. I guess the short answer is it is largely taphonomic and preservational as to why the Eocene. I mean, it is true. The Eocene is the first geological epoch in which you find something that is definitely a percodactyl. Paleocene has representatives of modern orders. There are carnivores in the Paleocene. There are primates in the Paleocene. There are rodents in the Paleocene. But most of the 20-ish mammalian orders are clearly recognizable a bit later.
Well, I might, as a geologist, I might just add to that the end of the Paleocene, beginning of the Eocene was one of the warmest periods in modern history. I mean, it was a lot warmer than the global warming we're getting now, so maybe life was a bit easier for mammals.
Indeed.
And also, if you just go back a little bit to the end of the Cretaceous, just 5 million years earlier, of course the dinosaurs had all got wiped out, and a lot of other stuff had been wiped out by asteroid impacts and big volcanism, so it left space for other things to radiate and fill, and maybe that's where mammals got our chance. You know, so maybe we're here because of a meteorite impact. Otherwise we'd be dinosaurs. So, you know, those are other factors playing into it, aren't they, as well as the preservation potential of actually being able to find what we find. Yep. Now, there's lots of hands going up, but I think you were first.
I talk about this kind of stuff primarily in church environments, and what I've discovered is that many Christians don't have a complete understanding of a good understanding of evolution, including the randomness of it, the mutations. And so I find that often churches or Christians often adopt a form of evolution and then say that God guided it. Whereas, from what I understand, a healthy scientific view of evolution has a random mutation part of it. And I found that when explaining that to people of faith, that is one of the more troubling aspects of evolution. And I was wondering if you could speak to that randomness and that the chaos in there.
There are a couple of really great points in what you've articulated that are worthy of extensive discussion. First of all, randomness. The idea that natural selection, or most processes in biology, are truly random along the lines of a vast series of slot machines, right? One reason why I think that Expelled movie is awful, because that's exactly what they do. They present evolutionary biology as if it were tantamount to taking a series of a million dice and rolling sixes all at once. I mean, that is that is an awful misportrayal, because as a matter of fact, selection is a biased process. It is not a random process. Now, you're correct, of course, to say that if you define randomness, and Elliot Sober, thanks to the Faraday Institute, was here some months ago and made this very, very good point. Random, in an evolutionary sense, really simply means that the mechanisms behind a change, say, for example, a mutation, have no immediate connection to the function that at some point in the future that mutation will serve, or that mutation will be relevant towards. So in an important sense, that is random, but it's not random in the same way that rolling a million sixes on dice is random. So that's one thing. To remind me, was that the bulk of the question? I think I've missed something that I've just forgotten.
I was actually kind of coming at it from the other side of things, in that people like the idea that God is in control of everything.
Indeed.
And that evolution leaves open space for whatever situation.
Sure. The other thing that I find very helpful is basically what you've articulated is this kind of notion that if it's natural, or indeed if it's random, it's not divine. And I would submit to you that that's a very parochial decision that you and I are bound to make. Because in terms of, if we look at our own creative capacity, indeed, if it happens in nature, or if it happens randomly, it's not the result of a human-like agency. I've never really understood why it's difficult from a theological perspective, and I'd be happy to be corrected by this, but why is it so difficult to take a step back and say, creation from a theological perspective, why can't that be random? Why is the mechanism of erosion that carves up the Grand Canyon, it's true, it's not purposeful in the sense that you and I do stuff in a purposeful way from day to day, but who are we to shackle the creator of the universe with this awful dichotomy between entropy and purpose? I mean, if you can, so I guess the answer that I would give to one of your parishioners would be, yes, there is a stochastic element in not only evolutionary biology, but most physical and life sciences. Why does that mean that this is not within the creative capacity of a God? And at some point that becomes difficult, because the critics will start saying, well, it's unnecessary, it's unparsimonious. And there's an element of truth to that. But that's another, I should let you guys do the talking about it.
Yeah.
I find it really helpful to read a description, Monsieur, that God has a plan but not a blueprint.
Indeed.
And the whole thing of chance enables life to be fertile and not sterile, and for that change and mutation to actually happen.
Indeed.
And that maybe it's actually a reflection, the benefits of God to actually have a creation that brings forth such potential and diversity.
Absolutely.
It seems to me too that the dialogue between the theory of evolution and those who look at things from a theological eye needs to be informed as well by, I think, an understanding of the history of how Genesis has been interpreted. A fundamentalist reading is actually comparatively recent.
Indeed.
And I think come through from the Adventist theologian, biblical scholars in particular, and then also coming through, obviously, in the United States very much.
Although I would point out that you mentioned the United States, and I'm compelled to say that the initial advocates of young Earth creationism in the USA was a Canadian.
That competition is not always taking place. That's right. And I gather too that also within Genesis 1, the Hebrew words for creator are different as well. There's something that starts almost from nothing, a bara, but also aslan in terms of shaping. And it seems to me that evolution is a wonderful way of actually shaping the growth of life and diversity. And is again a fruitful area of thought and dialogue.
Your trees don't give any mention to how soul might have arisen. And that's something that often comes up, so I was wondering what your view on that is.
That's a tough one. Soul. I mean, that, I suppose, I mean, I suppose one of the if many of you are avid readers of the Biologos website, and I do too. I read it too from time to time, and I think that recently was a particular issue in terms of, how do you accept this pretty good body of data in support not only of, primate ancestry of our own species, but the lack of a population bottleneck. I mean, from my perspective as a biologist, it is not reasonable to say that there was a time in human history when there were literally 2 human people on the planet. There is a signature in population genetics that we would pick up if that were the case. So the conversation then on the blogs with people flaming each other sort of goes along the lines of, well, there was no Adam and Eve, therefore the Bible is wrong. End of story. I'm in company. You guys have thought about this far more thoroughly than I have. I mean, but it is, I guess, my talk, so I can speculate.
Sure is.
I mean, at some point, I guess you have to somehow reconcile, what is it about a soul that is necessary to you to be a Christian, for example, or a Jew, or whatever faith you may be. And I'm still struggling with that, I mean, if a soul means that Adam and Eve lived in the garden, there were literally 2 people on earth, 6,000 years ago, then I don't believe in a soul. But, of course, that's a very narrow caricature, I think, of this large, these many, many ways that philosophers and theologians have been dealing with this thing. And I'm happy to, to hear some advice from my more informed colleagues on that. So I'm kind of evading your question.
Did you want to ask?
I want to follow on your question, actually. So, the KT event was when people think dinosaurs.
Cretaceous Tertiary, 60 million years ago.
Yeah, so when this asteroid hit the Yucatan,
Cretaceous in general.
so sometimes people say to me, well, that's how mammals were essentially nocturnal and hiding from the dinosaurs, and then they came out. Is that correct description, or were all these major groups that predate the KT event?
So, a couple of things. The first 2/3 of mammalian history took place before the Cretaceous Tertiary boundary. There were mammals for 140 million years before the KT boundary. Most of the trees that I was showing you, at least in terms of the evolution of pseudogenes, for example, in neoplastin, we're focusing on placental mammals. So the question is, were there primates, were there rodents, were there sloths, were there whales before the KT boundary? From a paleontological perspective, there is 0 evidence for the existence of any of these groups before the KT boundary. But of course, fossils, they're always a minimum estimate. No one's going to pick up a fossil and say, that's a whale, but I don't think whales originated for another 5 million years, obviously, But you can't say the opposite. It's always going to be a minimum estimate, and no right-thinking paleontologist is going to insist that primates originated in the lost Cabinian of the Wasachian of the early Eocene, because that's how old Shoshone says. That they were around before that. So in terms of the timing, it's pretty clear that at least some lineages of modern orders of placental mammals predated the KT boundary. It is equally clear that there was a tremendous diversity of mammals during the Mesozoic, prior to the KT boundary, including the famous dinosaur-eating mammal Rapinomamus, including things like Volaticotherium, which if you saw it today, you might think it was a gliding squirrel, but it wasn't. I mean, there were mammals that were the size of an obese raccoon back in the Mesozoic. There were animals that were, not much bigger than a paperclip. There was a substantial diversity. And what else did I want to know? Oh, yes, so you mentioned the asteroid impact. I think, Bob, you can comment on that far better than I can, but in fact, quite recently there was a paper about apparently the Deccan traps were active, around that time as well. And I guess the thing about the asteroid impact that makes it compelling is that you do find global traces of iridium, which is an element you don't find terrestrially. It only comes from meteorites. And even though that's a Yucatan Peninsula, New World impact site, you go to Italy and you will find iridium spikes around the KT boundary. You will find a fern spike above the iridium spike, because that's apparently, if you have a global nuclear winter type scenario, which is perhaps what happened in the centuries following, or at least decades, the first thing to grow would be lots of ferns. Right? So clearly the impact influenced things. It wasn't the only, I would say. I mean, nothing is ever that simple, really. Well, almost nothing. But I personally don't find it is quite reasonable to say that the KT bolide impact clearly influenced the terrestrial biota. The last thing I would say on that point is that dinosaurs are not extinct, because birds are nested within the clade of theropods. If you want to call a stegosaur a dinosaur, if you want to call a T-Rex a dinosaur, you have to call birds dinosaurs. But that wasn't really
Well, I mean, since you asked, I can say something about the KT boundary, because I wrote some papers on it a while ago. There were two things going on. There was the Deccan traps, which produced 10 million cubic kilometres of igneous rock in a tiny, short period geologically. Massive environmental impact. But the interesting thing about volcanic eruptions, like the Deccan traps, is they don't all happen at once. And as we've got better and better at dating it, it's clear that there were half a dozen really serious eruption events on land, so it affects the whole of the atmosphere and the whole of the world through that. They lasted relatively short periods, but they happened about every 50,000 years over a period of 500,000 years. So there's maybe 8 or 10 events. So each time you do it, you impact the biosphere time after time. So there's if you survive one, there's another one coming, then another one, then another one. And then right in the middle of that, there was this massive asteroid impact, which was probably a once-off, or maybe 2 or 3 impactors, one after the other. And I think it's the double whammy of the volcanism plus the impact, which did so much damage, and actually made 95% of species go extinct. So you had a sort of cleaning out of the environmental space, the biological space. And there were mammals, as you said before, and some dinosaurs after. And I think the dinosaurs were dying out anyway before the KT boundary for various reasons. But suddenly you had this massive space to radiate into, and I guess that's where mammals made it. And the dinosaurs didn't after the KT boundary. So in that respect, then we're here because of that, but their progenitors were there before. So it's a really interesting sort of interplay between the space available for biology to occupy and external physical factors. And the KT boundary is the one we know about, but there's half a dozen other similar cases further back through geological time where various species were wiped out and other things then had the space to come in and dominate, due to different factors maybe in other cases, maybe not impacts. So life is always slightly more complicated than you think.
I get the impression that God may not play dice, but he plays games.
Yeah.
That's fun.
So do you say that by way of? I mean, it sounds to me that the next, he plays games, comma. What kind of monster would use selection as a means to create biodiversity? I mean, that's a critique of the sort of theistic evolution perspective that I find most reasonable. That's a critique of that is pretty serious and something that is worth. But I'm putting words in your mouth. You didn't complain about nature.
Are games good or bad? Good. Good games.
Okay.
Problem solved.
Yeah.
I'm not quite sure how to formulate a question, but this business of supernatural and natural, I find from what you've said and other speakers have said that there is within this process, and for example molecular biology kind of confirming many aspects of the evolutionary theory. I find it rather wonderful, and it kind of inspires me, and in a sense I would say that's kind of, because it's beyond my comprehension, it's supernatural, and my response to that is awe and wonderment and praise. But I think the problem is, as a species, we want God to do things for us on an individual level.
Mm-hmm.
And then that comes to fiddling with the laws.
Mm-hmm.
And then I get run into problems because I don't understand why that would need to happen. And it makes God a bit capricious as well, actually. I don't know whether you have a view about that.
Well, again, it boils down to this view of, does a given contemporary religious belief system require an anthropomorphic God? Does the means of acting in the world require something that you and I would look at and say, no, that's not random, no, that's not natural, therefore it's God? I find that a very artificial. Phrased differently, I don't think that's a reasonable demand, even though there are mainstream components of Christianity that seem to insist on this sort of literal mode of activity of a creator in the world, at least in my view. But I sense what you're saying is that perhaps we're not so far apart on that point, that appreciating, say, for example, erosion as a creative means by which the Grand Canyon was created, in my mind, doesn't deny at all the agency of a creator behind it, in the same way that the means of selection or a number of other mechanisms that we know about biology, that doesn't deny the potential activity of an agency in them.
Do you have a last question?
Yeah. Raise a point, it's just about the population of the Earth. What is your view of the idea that most people are related to a very small group of people that came out of Africa sometime in the height of the last ice age, when the Sahara wasn't there to stop northward migration? So in that sense, everybody is related to this very small group of people.
I agree with one qualification, that it's a bit older than the most recent ice age. I mean, I think the data seem to indicate that about 150,000 years ago,
that'd be back in the ice age.
There would have been, a bit older than that.
Yes.
This is not at all the meat and potatoes of what I do on a daily basis, but I think from a population genetic standpoint, there is no question that everyone in this room, if a person of British heritage compares their genome with someone from the subcontinent, with someone from Australia, with someone from East Asia, they will have more in common with each other than a Pygmy tribesman and a Bantu tribesman who live next door to each other in Nairobi. There's more genetic diversity on the African continent among humans than there is anywhere else, and that is telling us something about the pattern of human migrations out of Africa. So there is, to my mind, no question that this idea of the peopling of the earth resulting from one or few out of Africa style migrations within the last 500,000 years, I think that's a pretty solid idea.
Right. Just another point. I mean, 60,000 years ago is sort of, I think, where most people think genetically the small population. There might have been a few, but that sort of period, I think it's, yeah.
Thank you.
Good. Well, I'm glad we're all related. Nice to know. So good moments to stop and thank Robert again very much.